So I'm Luda. Thank you very much for inviting our group HPC Bio to present the ongoing work for human genomic variant calling on BlueWaters. The slides will come up in just a moment, but I figure I'd start talking at you right away. HPC Bio stands for High Performance Computing for Biological Applications, and we are part of the Computational Genomics Initiative at the University of Illinois, which is a collaboration between biologists, bioinformaticians, computer scientists, and engineers. And we're interested in figuring out what is the best, most appropriate hardware and software for genomics. So we benchmark software to figure out where the software bottlenecks are. We rewrite the software. We also benchmark hardware performance. We try to project what would be the best hardware architecture for genomics. And ultimately, we're interested to figure out what is the best, most appropriate computer for high-performance genomic applications. Thank you. So needless to say, this is a highly collaborative field. A lot of the work that I'm presenting today was done in collaboration with Dr. Ravi Iyer at the University of Illinois and his students. Also, NCSA has a big involvement, as well as the BlueWaters and even Cray. Because the time is limited and we only really have 15-20 minutes, I have decided to focus the talk on discussing the motivation and context for human genomic variant calling. Why is it important? Why should we bother? And why should we be running it on a computer the size of BlueWaters? Then we will discuss the work in progress, some quick results, and finally, and importantly, the outlook. Why, for how long will our results matter from today? So let us first start with the justification. What is genomic variant calling, and why do we think that it's important? So genomic variants are simply differences in the genomic code between one individual and another. And if you imagine DNA is a very long string of letters, the genomic variant is simply the difference in those letters. It can be a substitution of one letter with another. It can be, like here in the red, it can be a duplication, an insertion, or a deletion. And this pertains not only to single letters, single nucleotides, but also to regions of DNA, sometimes whole chromosomes. These can be important when they affect phenotype. Sometimes the variants are silent, sometimes they affect the phenotype in an interesting but ultimately unimpactful way, such as this blonde but African child. And sometimes they affect phenotype in ways that are really important for our society. For example, in agriculture, the genomic variants can be important for plant biomass, or animal biomass, or animal productivity in terms of milk production. They can also be important in human health and disease, as genomic variants have been found to be causative of certain diseases. So how are genomic variants identified? Taking the human health area as the question, of course, if you are looking at a disease that is very well studied and a patient presents with a very well-known disease phenotype, you know which variants to look for. So that's where you look. You run a panel, which is a set of genomic locations where you expect the variants that are known to be found. And then you identify them just in those narrowly defined locations. This can be done mostly in an experiment, and whatever computational needs to be done is not really a challenge. However, if a patient presents with a highly recalcitrant cancer, for example, such that it resists treatment, or if a patient presents with an unknown disease phenotype, or a rare disease, this is especially important for childhood diseases, you have to find genomic variants de novo. You don't know them ahead of time. And in those cases, it is necessary to perform whole genome sequencing, which is you take a sample, extract DNA, chop it up into little pieces, and then you try to map them back against the whole human genome reference, which is an average representation of whole human genome, how it's supposed to look like. And by mapping the short pieces of DNA from the patient back to the reference, you can tell where the differences are. And then you construct a short list to identify whether those differences or variants are causative of the disease that you're looking at. So for a single patient or one individual, This is not a tremendously computationally challenging problem. It could be done in a few days to a couple of weeks on a moderate cluster. It's happening right now. We're used to it. It's not a BlueWaters kind of challenge. So what is? It appears that we have entered the era of personalized medicine. This is something that the scientists and the medical community have known would be happening for years. And now the government has caught up as well. Mr. Obama has announced earlier this year the Precision Medicine Initiative. He actually has been on a campaign to promote personalized medicine for some years, and finally it is official. In his address just earlier this year in January, he said that he wanted to bring us closer for curing diseases like cancer and diabetes, and to give all of us access to personalized information that we need ourselves and our families to be more healthy. He wants the country that eliminated polio and mapped the human genome to lead a new era of medicine. So this is big. When you see words new era, that means this is something big and different. One that delivers the right treatment at the right time. So NIH defined precision medicine as an approach for disease treatment and prevention that takes into account individual variability of genes, which is what I've been talking about today so far, as well as environment and lifestyle for each person. That means, literally, that we are now already entered the new era, the new time, where every person might be sequenced for variability in their genes, which is Gattaca. You guys, this is no longer science fiction. Gattaca is here, we're living it right now, and we had better be prepared for it. So for this reason, my case of choice for studying on BlueWaters is simply, what if we had to genotype every baby being born? In the state of Illinois, on the census of 2012, that used to be about 500 babies per day that were being born. So we say, what if we had to run variant calling workflow on 500 genomes every day? Is it a computationally challenging problem? I'm going to tell you in part 2. It turns out that it is. It is not just computationally challenging, it is a big data, big compute problem on a sustained basis. So what we're talking about is 500 babies being born in the state of Illinois every day. Baby comes out, sample gets taken of blood, throw it on the sequencer, we get the sequences back, push it into a supercomputer. At the end of the day, we have to get the result. What is the result? Well, does he have a nerve condition, obesity? Does he have early fatal potential and all that good stuff? That's what we're aiming for every single day. 500 babies, get it done. So according to our calculations, it is a big data, big compute problem because if you take just the input data alone, it's fairly easy to create a situation of 200 terabytes per day, which by itself is not tremendously large, but if you think about it, you run it for a week, you're up to a petabyte, just input data, without any processing whatsoever. You start processing the data, balloon out of proportion. You create about a petabyte, you create about a petabyte of data per day. Now, these data might be deleted eventually, but they still have to be created, stored somewhere, saved, moved around, processed. So this is a petabyte per day on a sustained basis. Besides, we're also generating hundreds of thousands of files during the intermediary phase of computation. Computational cost is also high, hundreds of thousands of node hours per day. And this is the reason why I call it big data, big compute problem on a sustained basis. And this is the reason why we need BlueWaters and the BlueWaters team, because we really want to know what kind of computational facility can sustain this kind of throughput. So our goals on BlueWaters are set up the workflow, which is done, prove that it can work on some test cases, which is done. I have successfully run it on individual genomes, 100 genomes several times. Now, a 500 genome case I can run, but it will eat up a third of my allocation, so I cannot do it lightly. We want to profile the hell out of the performance of this workflow, both for hardware and for software. After the profiling, we want to figure out whether there are any performance bottlenecks and eliminate them. And the reason why we want to do this is, well, number one, when Mr. Obama decides that it's time to genotype every baby being born, we want him to stand up and say, Illinois is ready. Everybody sequence your babies and send them to BlueWaters. because they have it made. So that's number one. Number two, we want to be able to, more prosaically, make recommendations for what kind of computational facility should the United States government be building in order to analyze these kinds of data. So we have two kinds of challenges. One has to do with data management, the other has to do with workflow management. It's fairly easy to foresee how they come to be, but let's go into detail. In terms of data management, it's fairly obvious that due to the size of the total data footprint and the total number of files that we're analyzing, everything has to be automated. So we need to auto-check for integrity of input, intermediary, and output data, making sure that every step of the workflow performs correctly. We need to possibly auto-stream data from the sequencing facility into the workflow, auto-stream into the archive of some sort, auto-zip, auto-compress, whatever, and potentially also auto-stream to the recipient. Now, these are solved problems in such fields as particle physics and astronomy, and we're not looking to reinvent any wheels. We just need help to learn, borrow, and adopt these solutions for our problems. Of course, this is where the BlueWaters team is invaluable because they're there to help all the time. We also have worked to identify potential bottlenecks, which have to do always with I/O, because we're handling big data. Potential bottlenecks involve uneven file distribution, simultaneous file access, saturating I/O at certain steps of the workflow, potential impact on the metadata servers. The good news is that BlueWaters has turned out to be extremely well set up to handle this kind of load, and we have not been able to create any problems tremendously. So that is good news. After a lot of profiling, we have identified some corner cases, and now what we're working on is designing some stress tests in order to determine how far we can push the system before anything can potentially break. So needless to say, the BlueWaters team have been invaluable. They have held our hand every step of the way. They have helped with the profiling, the finding of solutions, implementing solutions. So if NSF ever wants to have those paychecks justified, they should come straight to me. In addition, we have done this work closely in collaboration with the lab of Dr. Ravi Iyer and his students. Finally, I get to the workflow management and scheduling problems. And here I'm showing you that the workflow is comprised of 3 big steps, each containing multiple sub-steps, which do not necessarily finish in under 24 hours of wall time, which means that we have to break up every block of computation into individual jobs. Now, this is a very well worked out, very well published, trusted, and utilized workflow throughout the globe. So this is actually the standard in terms of the actual software that is being used. However, because of the complexity and the multi-step nature of the workflow, it tends to create a lot of intermediary jobs. So for example, here I'm demonstrating that in the middle of the workflow, it's very easy to generate tens of thousands of individual jobs, which generally is not a good idea to submit to a scheduler. Once again, the BlueWaters support team came to the rescue and introduced us to an MPI launcher, which is an MPI wrapper around our multiple jobs. We submit a single job for multiple nodes, hundreds, thousands, whatever, and then within that space, it places our individual tasks on their own nodes. So that works very well. Here I'm showing a Gantt chart for an experiment with 100 whole human genomes. It completes in under 30 hours, which I'm telling you is very good. You can see here clearly the first step in the computation. Once it's done, the second step is released. And then once that is done, the dependent jobs in the third step are released as well. So you might say, very reasonably, Luda, why create all these problems? You just need to write better workflows. Make it all be a single job, eliminate the intermediary files, maybe try to encode your data better so you wouldn't have to lug around all these bits and bytes. And I completely agree with you. Indeed, there is an effort to do precisely that, making big data be small data and making big compute be small compute. Now, this effort is sort of between the private sector and the academic enterprise. There is an effort to create what is called ultra-fast monolithic workflows, By ultra-fast, we mean they boast, producing variants on the whole human genome in under an hour, as opposed to 30 hours, like we do. It is monolithic, which means you only submit 1 or 2 jobs that completes under an hour, as opposed to tens of thousands that I showed you just now. So this would completely eliminate all the problems I showed you so far. There is also academic effort on a deeper level to actually change the way we handle the data. Instead of keeping the letters, encoding it in binary, compressing data, computing on compressed data, changing the way output is created. So this is fantastic. You might say, Luda, why don't you just wait a couple of years? Just go do something else. There's plenty to do. Don't worry about it. People will come up with solutions, and it will not be an HPC problem anymore. So there are 2 issues here. Number 1, I have a problem with that attitude. I feel like things get done when people do them. And I'm one of those people, so I'm not qualified to do any of this, but I can do whatever I showed you so far. And number 2 is that the medical community is actually extremely conservative. And once they have been introduced to something that they trust, something that works, something that has been validated for years, it is very difficult to convert them to something that has not yet been tested. So this work I showed you just now is extremely new. We actually have made collaborations with some of these groups. Some of them were very pleasant and successful, some of them maybe not so. But this is still very much a step in progress. It is not here. Solution is not here, but Gattaca is here. So we have to be ready now. And I will show you why. There's actually a production case that landed into our lab not a month ago. It is an international collaboration between the Consortium on Human Health and Heredity in Africa, the Sanger Institute, and the Baylor College of Medicine. They're sequencing 2,000 genomes because they're trying to establish the genomic diversity of people across the African continent, which should help them figure out why Africans tend to be predisposed to certain diseases, how to help them overcome those diseases, and also help us understand evolution. So this is a very important project. And most of the sequencing and analysis being done in Sanger. However, Baylor is sequencing 350 genomes, a fairly high coverage. So we already have an ongoing collaboration with the Africans, and our collaborators called up once and said, can you back us up? We may not have the infrastructure to handle this influx of 350 genomes. We have to analyze them in the span of 2 months. This is a big data, big compute problem. And because we have done everything that I've showed you in the slides upstairs, we were able to say, yes, we have tested this workflow. It is functioning. It is ready. Just send us your data, and we'll get it done. So this is the reason why we were bothering to do all this research. Because if we don't, we can't say yes. And yet the problems are here. So I would like to thank you very much in order to make this possible, making it possible for us to say yes. It's a highly collaborative endeavor between Institute for Genomic Biology, of course, CompGen, NCSA in a big way, of course, BlueWaters support team, and Cray also helped us with performance. Thank you very much.
If I understand you correctly, your effort is to introduce a workflow to basically pre-screen every newborn for pre-existing genetic conditions. If that's right. And so I'm a bit stumped how this can be, scientific or an NSF approach rather than an NIH one, for example.
I'm going to restate your question. Please correct me if I'm wrong. Your question is, do I believe that the problem of pre-screening 500 babies every day is a problem that's more appropriate for NIH rather than NSF? It might. Everything that has to do with human health is generally the provenance of NIH, and that's correct. However, the provenance of NSF is to establish the science that makes it possible to do research in human health. Now, the work that we have done is computer science work because we studied performance on a large supercomputer of a workflow which is a piece of software. And that is why our work is appropriate for BlueWaters and NSF.
Other questions? Okay, thank you very much.
Thank you.